Hello everyone. Thank you very much for joining us today. My name's Louise Tratt, and on behalf of the British Neuroscience Association, I'd like to give you a warm welcome to this webinar, Advancements in Multi-Electrode Recording Techniques in Neurophysiology, from Wire Probes to Neuropixels, which is hosted jointly with our annual supporters, Scientifica. Just before we start, and while people are still joining us today, we have just a few housekeeping rules to go over. We'll be using the chat and the Q&A function today, so feel free to say hello and post comments in the chat. But if you have any specific questions for our speakers, then please make sure you use the Q&A function for these. You should see both the chat and the Q&A buttons at the bottom of your screen. There'll be a Q&A session after the talks today, so you'll have the chance for our panelists to answer any questions you may have. So feel free to post your questions at any time during the talks, and we'll pick them up at the end. You have the option to ask these questions anonymously if you wish to do so. I'd like to take this opportunity to remind you this webinar is being recorded with the permission of our speakers, and it will be available to watch on demand on the Scientifica and BNA websites. And finally, just a reminder to please adhere to the BNA Code of Conduct for webinars and engage respectfully online. Okay, so without further ado, it gives me great pleasure to hand over to Dr. Chris Schofer, who is US Product Specialist at Scientifica, and they'll officially kick off the webinar tonight. Thank you.
Hey, thank you so much. It's great that everyone was able to join us today, and I appreciate your time. Get this started. Is this showing up okay, I hope? All right, so thank you again for that introduction. Today we're going to be chatting quite a bit about in vivo electrophysiology and multi-electrode recording techniques. And so here's a brief outline of our talk today. So I'll start with a very brief introduction of extracellular recordings, discuss some benefits on why people may want to research with multi-channel electrode recording techniques. I'll review some basic equipment lists and some considerations when you're planning these types of experiments. And then lastly, I'll give a brief overview of various categories of some multi-electrode recording technologies. And then after that, we'll hand it over to our keynote speaker, Dr. Sylvia Schroeder, for a feature on Neuropixels technology. All right, so as a brief introduction, extracellular recordings in the brain, we are, by the way, I should note that all of this is primarily focused for small animals. However, it absolutely applies to invertebrate species, non-human primates, anything with the central nervous system and neurons in a group. So there are a few different types of extracellular activity that one can record in vivo. One, of course, being action potentials. We know them, we love them. In the literature, you may also find this referred to as spiking activity or firing a single unit activity, a unit being a single neuron. And we can also record multi-unit activity. So if we have one sensor, it's surrounded by a bunch of neurons, and we're able to record the spikes from many neurons at the tip there. And of more interest to many is the local field potential. And so these might be these sort of continuously sampled traces that vary. You see these temporal fluctuations in voltage across time. And so one is able to measure the local field potential at various points in the brain. For example, here's a nice image from a review several years ago from the Bujaki group, demonstrating that, yes, we have the action potentials. You could record those intracellularly, and you see these same patterns mimicked in the local field potentials. And one major perk of modern recording techniques is that by filtering in the frequency domain, you're able to acquire both the local field potential, so these oscillations, these fluctuations in voltage over time, by applying a low pass filter. So maybe 2000 hertz and below, something like this. Or if you were to high pass filter the same raw data, you're able to isolate the spiking activity, the action potentials. That is the multi-unit activity here. So why switch from, for example, recording a single neuron, maybe a patch clamp experiment ex vivo or in slice or in culture, to something multi-channel and a little more complex? So here's a figure of PubMed new articles coming out per year, new publications with neural circuit as a keyword. Notably, not all of these will be for will include electrophysiology, but it's undoubtable that neural circuits and examining the central nervous system at more of a systems approach, right, at a larger scale, is becoming increasingly relevant and popular in modern research. And so the major benefit of studying in vivo is that you get to study the central nervous system intact as it is, as the subject, uses their brain in normal ways, rather than in slice where you sort of isolate just microcircuits and you're cutting out a lot of the standard inputs. You can also, unlike slice electrophysiology, you can acquire multiple recordings from a single subject over the course of days. By its nature, right, having multiple electrodes in place, you can record multiple neurons, and by applying more than one probe across the brain, you can record activity from interconnected brain regions. So maybe brain regions that you suspect may be communicating with one another in real time. And this gives you a much broader understanding of the brain and your PET circuit as a whole. And then lastly, in vivo multi-channel electrophysiology, when in preps where animals are awake and behaving, this gives you the major benefit of linking neural time series events with behavioral actions in real time. So moving on to a few general considerations, if you are not currently using these types of research approaches in your laboratory, but you're thinking about it and not really sure where to start. One thing, of course, is starting with your animal subject, presumably animal, mice or rats or guinea pigs or ferrets or anything larger. It's something to consider is how much hardware can your animal carry around on its head unencumbered. This is mostly relevant for mice who can maybe carry a couple grams at most on their head without, while still being able to lift their head in their home cage, as in terms of chronic implants. But if you were looking to record with a larger amount of hardware on a small animal, you may consider acute recordings where the animal is anesthetized and you bring the equipment to the animal temporarily, and then it can go back to its home cage without carrying a large head cap around. For folks that are looking to do multiple recording sites, so within the brain, you should also really consider what the implant surgery will look like. So there's a limited amount of space on the skull. So you need the space for craniotomies to place, imagining where your electrodes will go inside of the brain. You don't want anything clashing or running into major blood vessels, ideally. Also for folks who do head-fixed awake behaving experiments, you may need to insert a head-fixed post, essentially, and then skull screws to keep everything stable in place. We don't want any of that equipment coming off of the animal unexpectedly. Something else to consider in this stage is where to place the ground electrode. So for acute anesthetized recordings, this is a bit easier you can basically put a small alligator clip on the animal's forepaw or some other kind of similar extremity. It's even better if you maybe soak a piece of gauze in saline or something conductive like that to make sure that you keep the ground connected to the animal stably throughout the entirety of your experiment. But for chronic implants, so folks that do awake, freely moving, there may be a designated skull screw that's soldered to a pin that's set as a designated ground. And there may be an implanted electrode that is also serving as a designated animal ground. This is right to complete the circuit here, close the circuit. And when deciding where to implant the ground circuit, the ground electrode, you're going to want to choose a location in the brain that is relatively free or has very low amplitude neural oscillations in the local field potential, like we were showing. So something like a very thick white matter bundle, something like the arbor vitae in the cerebellum, or maybe the corpus callosum. Those are generally popular choices. Something else you might think about is the other types of streaming data sources you want to synchronize with the neural data. So for example, video tracking, maybe pupil dilation, or any kind of behavioral apparatus. And as research continues, the closed loop manipulation experiments are becoming increasingly popular. So things like having, when the brain state reaches a certain point, or the animal does a certain behavior to trigger optogenetics along with your recordings. So these things are one, to think about. And then lastly, after all the recordings are complete and ready to terminate the animal, finish that experiment, ideally people would really like to check the histology, right? See where your probe has gone in the tissue. And there are different ways to do this. You can either hit the surgery stage, dip the probe essentially in some kind of fluorescent reagent to mark it quite clearly. During an anesthetize, the animal you could apply a current to create an electrolesion, or something as basic as just a Nissl stain here. But as you can see, that depending on which type of technology you're using, the tracks are really quite small and can be easy to miss. So some general equipment, of course, you're going to need, so on the data collection side, you're going to need a data acquisition system. This usually kind of looks like a large box that sits on the table with some inputs, and it would feed into your computer workstation sometimes it feeds by a fiber optic cable. There are different ways to transfer this high bandwidth data live, but working backwards from the data acquisition station, when you go to purchase this, you will need to consider how many channels, how many electrodes in the brain you'd like to have, and consider if that system can be expanded in the future. Can you add more channel banks if needed? All right, so the head stage preamplifier is this piece here. And what this does is it's the closest to the brain, right? The voltage signals are arising from the brain, going right into something that would digitize them and preamplify them, essentially. So we're working from very small voltages. Of course, you'd need the electrode, the bit that would go inside the brain, and that has the sensors on it to transduce the voltage signal. And between the head stage and the electrode is something called an EIB. It's an electrode interface board. They can look many different ways. It's essentially a solid panel, a plank that can serve as your input-output, something to attach the head stage into without wiggling around the equipment that's inside the animal's brain. Occasionally a multi-channel impedance tester, and also data storage. So also for awake behaving head fixed experiments, you may need a head fixation system. This is a nice low cost open source option we recommend called Rivets, where you can 3D print essentially your own head fixing device. And then for freely moving experiments, there's also, so the animal is essentially tethered. There's a tether going from the head stage out. And when the animal spins in circles, you don't want that tether gathering up tension. So typically the tether goes into a commutator, which then feeds to the data acquisition system. And the commutator's job is to unravel the signal tether as the animal moves around. You also, of course, the behavioral apparatus, make sure it's either constructed of something that won't acquire static electricity, static charge, or you can spray maybe a plastic system down with an anti-static spray. There are ways around it. Okay, on the surgical side of things, you will definitely need a well-maintained set of surgical hand tools. And I say that well-maintained, this includes very fine forceps. So something to, once you remove the piece of skull, do the craniotomy. There's also the durotomy, right, where you need to remove just a small portion of the dura to access the brain and drive the probe in. And so this requires very fine, well-maintained tweezers. It also would definitely benefit to have either a digital microscope or a high magnification stereoscope, so you can do the surgery under microscopy. Of course, you need a stereotaxic apparatus, something like this, to fix the subject's head in place while you're doing the surgery. You'd need a Dremel style drill to accomplish the craniotomies in most cases with a thicker skull, certainly for rats and larger. We also recommend a pulse oximeter. So this is something to monitor the vital signs, as these implant surgeries can last several hours depending how involved they are. And so an animal being maintained under anesthesia for this amount of time, it's good to have kind of a monitor, just check in with it every now and then. And then of course, you will definitely need an ultra stable micromanipulator, something to drive the probe into the brain to your target. I'd like to take a little moment here to plug some options that Scientifica can offer for this. Ideally, you'd like something that is electrically silent, especially if you're doing acute recordings, right? If you're going to be recording right there in the surgery suite. And so something that you can essentially pre-program in, and it will have a motorized drive. So you click go, you set up your probe as you need. You essentially click go and it will drive reliably to your end destination. And it is known that generally speaking, the slower that you drive this external hardware into the brain, the better results you'll have. So you'll have less inflammation, less gliosis, and general specs, depending on the type of hardware, depending on if the implant is going to be chronic or acute, and the tensile strength, so how hard the implant is. The speeds can vary, but typically somewhere between 10 and 100 micrometers per minute, so a very slow drive. It's a little tricky to do by hand and keep it consistent in movement, so we highly recommend a motorized manipulator for this part. Okay, oh, and as I mentioned, right, if you're taking an acute or anesthetized recording, you would have all of the data acquisition there with you in that surgery space. And it's also, don't forget to ground, the stereotax frame, the table, essentially anything that that could take on some interference, some electromagnetic or interference, I guess, from the environment, because you don't have a Faraday cage typically to help block that. Okay, and I would also like to just do a brief overview of a few different modern technologies. I want to note that this is not an exhaustive list. So we're just talking about the different types of sensors, the probes that can go into the brain to get your signal. So there are new ways to record and manipulate neural activity in vivo. It's being tested and developed in laboratories worldwide. So this is by no means exhaustive. So starting from a most basic and budget-friendly, most widely accessible version, this is personally what I worked with when I was doing my PhD, is hand building some microwire bundles. And so this is typically some fine wire, it's insulated, typically either tungsten or stainless steel type of wires. Some pros, right? Of course, it's very budget-friendly, easily accessible. You essentially twist them together, use maybe a heat gun to kind of keep it whole, and using a scalpel to cut the tips. And so the sensors, right, are just here at the tips. And of course, you can custom design these to whatever length that you need, whatever size. And it's easier to target multiple brain regions when you have just a couple flexible wires. Here's an example of one paper somewhat recently in Cell from Kafuchiras's lab where they were able to record from many different brain regions simultaneously using fairly basic passive probes like this. There are some downsides though. However, of course, you cannot isolate single neuron activity this way. You can, of course, get the multi-unit activity like we discussed earlier, but I'll mention Tetrodes in just a moment where we can isolate single unit spiking. There is a little bit of build time built into this, right, by nature. You're doing it yourself, so you take that risk. And there's also, because you're building it yourself, there is some rate and risk of quality control rejection here. And because these wires are somewhat larger in diameter, there's an upper limit to the channel count. You can't insert too much mass into the brain without hurting something. All right, next I'll skip to Tetrodes. So very similarly to how seismologists might locate an earthquake in space by getting kind of the different amplitudes and timings from different seismology recording sites, kind of combine that to triangulate a location in space. This is very similar approach used with tetrodes. So a tetrode, meaning four, you have four wires that are bundled very tightly together. These wires are much thinner than the previous ones we discussed. And because you have the four, right, you can get the 2D space. You need one more wire, then you have dimensions. So because we want to triangulate where a single neuron is, you need four. In 3D space, you need four wires. So, right, the main benefit of these is that you can isolate single unit spikes. There are a wide variety of spike sorting algorithms available to help separate those signals out. But small downside is that because as compared to the previous technique, you now for each location you want to implant an electrode, you actually have 4 channels to cover. So this may mean that you need to start with a larger data acquisition channel bank. And then I'll mention on these shuttle assemblies momentarily, but this can also add to build time. So if we wanted to take, that's just one tetrode, a hyperdrive is essentially taking many, many bundles of tetrodes and pooling them together into one implant so that you can maximize the number of neurons that you can record at one time. So you get more spike data per subject. Those mini shuttles I mentioned, so this is kind of what the hyperdrive looks like and what it looks like on a mouse here. But these shuttles, you can use a very small screwdriver to turn and individually drive a tetrode down into the brain by very small amounts. And so this allows you to track multiple neurons from one tetrode bundle over time. The downside, of course, here being it is a rather bulky head cap. There's some risk of the head cap coming off. And like the tetrodes, this one even does have a bit of a longer build time. And I did mention these are scalable. The hyperdrives are also open access. You can 3D print them yourself. Okay, moving now to active sensing probes. So these are kind of starting a newer technology, I guess, in the last couple decades, silicon-based probes and probe arrays. And so you can use these in either acute or chronic experiments. So you can, put the probe down and take it out the same day after you're done recording, or leave it in and keep it in place. Unlike the other ones, which would be handmade, these guys are typically produced somewhere in a company or facility, so you know that the quality control is excellent. You have customizable sensor layouts, so a lot of these companies are able to custom design probes for you and for your specific application, certain spacing, however you want to hit whichever brain targets you need. A lot of times the sensor and the circuitry is right there on the chip rather than being somewhere else kind of in the head stage or in the DAC. So you do get a little bit more, you do get a much cleaner signal rather. And then using a linear probe like this, you can do something like a current source density map. So neurons are little dipoles. And so especially in laminar regions, for example, the hippocampus or some cortical regions where the neurons are lined up in a row, it's almost like a battery cell in some sense. but you're able to reconstruct the current source sync dipoles for these types of electrodes to help help inform the exact location you're getting, and then the maybe juxtacellular recordings, or kind of help parse apart different spiking events and different classifications. All of these is great, but the downside to these, they are rather expensive consumable, typically costing several hundred to a couple $1,000s a piece, and they are easily damaged. For this reason, a lot of companies may sell kind of dummy probes for training. And I also wanted to just briefly touch on the Utah array and Michigan array. These are different styles of these kind of linear probes. So the Utah array, this is a 10 by 10 grid where the sensors are just on the tips here. And this is most common to cortical researchers, both for human research and preclinical research. And right, the spacing is rather fixed. And so it can be somewhat common to plunk down a couple of these Utah arrays, maybe in one location in the brain. And then the Michigan array are these kind of flexible ribbon-like cables with very dense sensor arrays on the tips here, not unlike the previous slide here. Essentially, this is, you can get more than one shank in the same device. And of course, these can be customized, different spacing depending on your needs. Okay, and then finally, this brings me to Neuropixels probes, which is the latest generation, nearly 1,000 channels, very, very, very densely packed. And the major selling point of this Neuropixels probes is that you are able to record hundreds of neuron spiking activity as if you had many, many tetrodes in one place here, all at once. And so the circuitry is right on the chip. You get very clean, reliable signals. Because this probe is so tiny physically, you have very minimal tissue damage. They're used most commonly in acute recordings, but the latest generation, I understand, can be cleaned and reused potentially between animals. And most importantly, there is a very large supportive worldwide user community with many online resources like a Slack channel. There's a main website, of course. There's a GitHub wiki, and there's just a lot of online material from previous workshops and training sessions. Really, the major downsides right now, as we see for Neuropixels, it is a pricey consumable. Because of the massive amounts of data you can acquire with these, this does also massively increase the need for adequate storage, data storage solutions, and more compute to work with the data. Finally, there are a few bottlenecks in the Neuropixels probes production and assembly and accessibility. Such that there is generally a somewhat long wait list to get your hands on some of these probes. But without further ado, I would like to turn it over to our keynote speaker here, Dr. Sylvia Schroeder, who is our local expert and does use Neuropixels regularly in her research. So Sylvia, I'll turn it over to you.
Yeah, hi. Welcome again, and thank you for inviting me. It's a pleasure to be here. I'd like to share my screen. And yeah, just 1 minute. Sorry. Yes, okay. I hope you can see the screen well. So yeah, I'm a group leader at University of Sussex. I have used Neuropixels previously in my postdoc and will be using it in my group now. And my goal today is to tell you a bit more about the technology of Neuropixels and its advantages, some tricks in using Neuropixels, what kind of data you can get with it. But I think most importantly, I want to show you what kind of resources are available. So Chris already mentioned there is a lot of resources there. And my goal is to make you a bit more aware of those and the community out there. So let's start with what's in there, what's the technology in the Neuropixels in a bit more detail. So we have this 1 centimeter long shank, and there are 960 sites along these 10 millimeters. At the same time, you can record from 384 channels wherever you want along this 1 centimeter. So you can record from those simultaneously, and it's very easy and quick to switch between any of these recording sites. So this means you can record from a lot more neurons compared to more traditional techniques. The distance between recording sites, so here these black pixels, is very small. So center to center distance is 25 microns. This means you get a much more detailed signature from each neuron. So when spike sorting, you and also any algorithm you can use can be much more certain in identifying each neuron. And it's also more likely that you can record from smaller neurons or neurons that are maybe further away, which could be missed by probes that have a bigger distance of channels. As Chris already mentioned, the architecture is very slim. So the width of the probe is 70 microns, its thickness is 20 microns, which reduces damage to the brain. And also Chris mentioned this, so amplifiers and digitiers are very close to the recording site. So it's included in the base. So about the 1 centimeter away from your probe. This means the analog signal has to travel only a very small distance before it's digitized. This means only there noise can be recorded. Once it's digitized, your data is safe. You only need to transmit ones and zeros to record on your computer. So that reduces noise a lot. And the probe is constructed so that it only weighs, the probe itself without the head stage, I have to say, weighs 250 milligram. So it's very suitable for even freely moving mice, where you would implant the probe on the head, so the mouse has to carry it all the time, basically. Only during the recording you would add the head stage and then the cable and would record the data. So here's an example recording to show you just how the data would look like. So these are only now 125 channels. So each line is a voltage trace, and it's a very short snippet, so you can see a little bit of the single spikes. This was actually recorded in a mouse with 4 probes at the same time. So they were all put in an area which I'm interested in, called the superior colliculus. So it is possible to really place even several probes in a relatively small area if you place them kind of cleverly and use the space available. And you see that some of the spikes are colored here, and this is to show you that this data was spike sorted, and the algorithm used was kilosort, in this case kilosort 2.0. And yeah, I showed you this so you can see that even very much overlapping spikes can be picked up by kilosort. And this is of course helped by the fact that a single neuron is recorded by many channels simultaneously. There's lots of tutorials available online on YouTube, which you can look at, and they talk about how kilosort works and also how you can use it, because it's a semi-automatic. There is a manual part to it, and in these videos you can learn how to use this. Next, I want to show you a bit of quantification, how many neurons you can record. Here you see 2 probes located in various brain areas. For example, visual cortex, hippocampus, thalamus. Here you see a recording. So this was already spike sorted. Each dot here is a single spike, and dots with the same color and the same height kind of come from a single neuron. Okay, so this is just what the almost raw data look like. So you can see you get lots of spikes from lots of neurons, but how many actually? So here's a quantification. Of course, the number of neurons you get on a single probe depends on which area you're in. So here's a list of several areas. You can pick your favorite one, for example, thalamus or sensory cortex. But yeah, on average, maybe, you know, there's several tens of neurons you can record at the same time, up to a couple of hundreds, depending on the area. And here in the second plot, you can see on how many channels on average you can see a single neuron. So again, yeah, this is more or less variable across areas, and you can see it's about 8 to 10 channels. So it's really good signature you get per neuron. And now I want to come to a typical problem of recordings in alive or awake animals, which is motion artifacts. You don't really hear about this a lot, maybe in papers, it's usually ignored, but it's definitely a problem that happens. And I think Neuropixels are really great in getting around this problem. So here is one recording from myself in the superior colliculus. Let's talk about this figure because you will see a few of those. So on the y-axis, we have the depth along the probe, and the x-axis is time. Each dot is again a single spike, and the color signifies the amplitude of each spike. So this data was not spike sorted, only spikes were detected, and then the dot was colored according to the amplitude of each spike. So you can see, you know, more or less across time, probably the same neurons with the same kind of spike size are constant, but not perfectly, right? So here, for example, you see there's kind of some jumps seem to happen, or maybe best you can see it down here. So that's relatively deep in the brain, and there's lots of instability. So if you had only kind of a stupid spike sorting algorithm that couldn't deal with these shifts along the probe, you would be lost. But even completely lost you would be if you only had a few channels recording from those neurons in the brain, and you would lose the neurons, and maybe you wouldn't even notice, because, you know, how do you know whether the neuron stops spiking or just vanishes from your probe? So this is really great about Neuropixels, I think. And here was a test to see how good Neuropixels and the proper algorithm with it can be. So here you see some data that was recorded in primary visual cortex. So you already can see here that there is, yeah, the motion here was really bad. So the probe did actually move quite a lot in the brain. Plus, then they actually moved the probe actively according to this zigzag pattern. And you see, of course, that the spikes move with it. After the correction, the data look like that. And yeah, once it's corrected like this, the spike sorting and identification of single neurons becomes much easier. So this is possible because the recording sites are so dense on the Neuropixel probes. And because someone very clever, Marius Pacitario, developed this algorithm called KiloSort 2.0, and then also later versions that can correct for these motion artifacts. This particular example was actually recorded on the next version of Neuropixels, which I will talk about a bit later. But believe me, even on Neuropixels 1, this works very well. And here's just a bit of quantification about stable units. So in the original data, you have fewer, you could detect fewer stable units than after motion correction, and the number of unstable units goes down a lot in the corrected version, right? Okay, now I would like to show you some data that I recorded that was very challenging, and I believe was probably only possible to record because of Neuropixels. So the challenge was to record from retinal axons in the optic tract. And you see here one brain slice, and in green, you can see the retinal axons. They're labeled with GFP, and also a faint trace of the Neuropixels probe that I used to record from this area. So the goal of this project was to find out how or whether behavior, for example, running, influences the visual signals that are coming from the retina. So basically, does the spiking of retinal ganglion cells change because the mouse behaves differently, right? And I have to say this, this whole data, I think, would not have been recorded, or yeah, it wouldn't have been possible to record without Neuropixels and without Keynote. Right, so how did we approach this? Here you see some data from one isolated unit. So you can see here the wave shape, the spike wave shape of this unit on several channels along the probe. And you can see it looks, if you know about spikes, this one really looks a bit funky. It has like several peaks and troughs. This is, if you go through the literature, that is somewhat expected if you record from things like axon. So a typical spike wave shape from neuron looks typically different than that. So this was one point that convinced us we are probably actually recording from an axon here. Then we looked at the cleanliness of the data. So here you see the autocorrelogram of the spikes of this unit. So it starts at 0 and then goes up to 1000 milliseconds. So you see up to 1 millisecond, spikes do not occur within a 1 millisecond bin. So this makes it unlikely that this is a multi-unit recording, because we know that a single neuron shouldn't spike 2 spikes within a millisecond, okay? So that was another quality measure. Then another measure to convince us and others that we are actually recording from retinal axons was a functional measure. So here we used the flickering full field stimulus. So the monitor was just flickering between black and white relatively fast. And here you see in each row the spikes in response to the monitor flickering at different frequencies. And this unit could follow this flicker very well. So this convinced us that this is a good reason that it's a recording from a retinal ganglion cell. And also this unit has a very small receptive field, which is another point convincing us that this is from the retina. And here you can see some data from the whole record, or from a long part of the recording. So on the x-axis is time. The first plot is the firing rate of this single neuron. And for a long time, we just recorded this unit during complete darkness. So the mouse didn't get any visual input. Then here you can see how the firing rate changes when we suddenly show this flickering stimulus. And then here we show the receptor field. So within each kind of paradigm, stimulus paradigm, the firing rates are stable, or they make a lot of sense, right? For certain frequencies, the unit spikes more than others. Here you can see a plot for the same data of the spike amplitude of this unit. So the y-axis is amplitude and x-axis is again time. And in color, you see kind of how many spikes. Basically, it's a histogram, a 3D histogram. And you can see, hopefully, that over time, the spike amplitude is actually changing. So there is probably some tiny little movement of the probe relative to the single unit, which makes this amplitude change. But luckily, the Keynote is able to figure this out and still track the same unit over a very, very long time. And here's another plot of the depth of the spike over time. So in this case, it didn't change much, but there are other cases where you could see depth changes, especially if you have bigger neurons, then they're relatively easy to track across depth, as we have seen before in the motion correction. Right, and here are some results from this data. So you see here the recording during darkness. you see in the first plot here, the running speed of the mouse, which is changing over time. So this is a time of about 40 minutes. And you see here the firing rate of a single retinal axon, a single unit. And we wanted to know whether there's any relationship between running and firing rate. It's not quite obvious here in this raw data, but if you plot a cross-parallelogram between these two variables, you can see that you can observe a positive correlation between running and spiking, and that is more than expected by chance. So it is significant. The reason why we chose darkness is that we wanted to exclude the possibility that there's any visual input that would change when the mouse is running around, maybe looking around. So visual input cannot explain this, this result. So it was very surprising to us. And yeah, it's a very intriguing result. Only possible with Neuropixels. Here's another, a second unit. And again, you can see here the running speed of the mouse. This was a different recording session. Here again, the firing rate of the unit. And again, this unit has a positive correlation. So in total, we were able to record from 25 units. It's a relatively low number, but it is, even with Neuropixels, very difficult to record from the optic tract, which is a very small, small brain area. And it is still quite difficult to get single units and to get it stable over long times, like 40 minutes to an hour. We also checked then actually whether the spike waveform changes with the running speed, which would of course be, hinting towards an artifact, and this was not the case. So the spike wave shape was the same during periods of not running and running in all these 25 units. So in total, we found now 9 units of 25 that are correlated with running speed during darkness. Right, now I would like to talk about the combination of Neuropixels recording and optogenetics, because that's another very popular method that is used in neuroscience. You probably all know about optogenetics, but very quickly, it's a method that can be used to activate and inactivate neurospiking by light. Basically, you use genetic methods to introduce light-sensitive channels into the cell membrane of the neuron. And so if you shine light on the neuron, depending on the channel, you can either activate, so make the neuron spike, or inactivate, so stop them from spiking. And here's a little mouse with a typical glass fiber that transports the light to the brain area where you want to have the effect. So if you want to combine this method with Neuropixels or any metal probe, there is a problem, because if you expose metal to light suddenly, it causes a voltage deflection in the high frequency band, which is the band where you record neural signals. However, there is a trick kind of to avoid this, namely if you, instead of having a step change in the light, you can use smooth changes in the light intensity, for example, raised cosines. And this even works for very high frequencies, which are usually used for optogenetics. So if you want like a 40 Hertz light stimulus to activate or inactivate your neurons, this is possible using these raised cosines without getting any artifacts on your probe, or at least in the high frequency channel. And here's some data to show this. Here's the different ways of switching on the light. So here is a ramp that suddenly stops, or these raised cosines that I mentioned before, or pulses. And here you can see what happens at the same time on the probe or in the voltage signal. So for these sudden changes, you have artifacts in the high frequency band right here, but you don't have any artifacts for these smoother signals, okay, like a raised cosine. Of course, well, you do have deflections in the low frequency bands. But if you're interested in spikes, then you don't need to worry about this, or it's a way around these artifacts. If you're interested in this, or also want to use optogenetics but have to use these kinds of step changes, then I really recommend you this video by Maxime Beau, who used this method and tells us about some tricks he used to get around these artifacts. Right, and here's some data that was collected in V1 during optogenetics. So here's the light stimulus. This is the 40 Hertz sine wave. This shows you kind of the firing rate of multiple neurons across the depth within cortex. So here the neurons are happily spiking away. As soon as the light comes on, many neurons stop spiking. After the light, they start again. And here you can see the effects on the LFP. So these raised cosines leave a funny, well, cosine pattern in the LFP. So probably you cannot easily use this LFP if you're interested in this. And here's some data from V1 neurons. So here here are single neurons recorded with Neuropixels while the light was shown onto the same spot where the Neuropixel probe was. Here you see several trials of the same neuron when the laser was off, but the visual stimulus was presented to the neuron. For the same neuron, when the light is on, the spiking stops completely. And I had no problems with any artifacts because I used these cosine patterns. And here are some other neurons. And here's also one neuron in V1 that was activated. So I forgot to tell you that this was in a mouse line which had the channels. So PV, it was a PV3 mouse line, and together with channel rhodopsin, so where the PV inhibitory neurons were actually activated. So this is the putative inhibitory neuron, which then inactivates all the excitatory neurons that you see here. Sorry for that. Right, so you can combine these techniques. I see we kind of run out of time, so very quickly, there are ways to determine your probe location, basically using histology by using dyes, as you can see here. You can just apply them onto your probe, and there's a very nice MATLAB program which you can get from these GitHub repositories, with which you can align your brain slices to the Allen Brain Atlas, and then even morph the atlas, or rather the brain slice, to make it consistent and matching with the atlas by using kind of some landmark dots that you place on the brain slice and also on the atlas. Then you can mark your probe location on the slice, and what you can get out is then kind of the whole brain and the location reconstructed that go through the brain. And also there's a straight line fit through the brain, which designates the location of your Neuropixels probe. And these, the software also gives you a vector with all the brain areas recorded from. And this you can then use to align to your recording. So you see where your, basically the depth of the brain areas relative to your probe. And just in mentioning, I want to say that yes, several labs and people have used Neuropixels to do chronic recordings. For example, here in mice, the data is very stable. Here you see firing rates across many, many days. So up here, 150 days after implantation. And the data was still very good. And here you see example waveforms recorded from day 153. And the waveforms look very large, so there was no quality loss, or hardly any quality loss in terms of recording quality. And there's several papers who have used this technique and also describe how they did it, so what kind of hardware they used, and also they developed hardware so that the probes can be recovered after the experiment and can be used in different animals. in a different animal if you're careful. Okay, and just to tell you, because Chris mentioned the price is relatively high, but what do you actually get in a package and how much does it all cost? So you get a whole acquisition system, which can take up to 16 probes, I think. You get this for, yeah, €2,500. Each single probe is about 1,200 euros, head stage 250 and cable 175. So just, I don't know, I got these prices end of last year, so it might have changed a little bit, but this gives you kind of an idea of what you have to pay. And just my last slide is on upcoming models. So this is a model with 4 shanks. It's called Neuropixels 2.0. So the new features are, this has 4 shanks with a lot more recording sites. So 5,120, more than 1,000 per shank. You can also get a single shank version, but probably, yeah, the 4 shank version might be more interesting. You can connect 2 probes very closely together on the same head stage as shown here. So you have more than 10,000 recording sites, and you can record simultaneously from 2 times 384 sites at the same time. Also a difference is that this base and the head stage are much, much smaller, and also they weigh far less. So even better for chronic recordings. The recording sites are much closer together, which helps even more for motion correction or artifact correction. And again, yeah, you could even record from smaller neurons if that's a problem for your brain area. These probes are, however, not available yet, but will probably come out next summer and will be available then to be used by anybody. And here's the list of resources I really want to give you. So here's the wiki page for the Neuropixels GitHub. There's a Slack channel, Neuropixels site. I have not at all talked about SpikeGLX, which is a software used to record the data, and it was specifically developed to record data from Neuropixels. And all the videos I mentioned on YouTube, you can find here. So they were all recorded during the UCL Neuropixels course 2020. So I recommend you check this out. And also I want to mention my funding buddies that made the work possible that I did, that I described here. Thank you very much. And maybe we have quick time for questions.
Fantastic talk. Yeah, so we do have a few questions that came in. All right, maybe we can start. So first, Bradley asks, is there an advantage to implanting a ground probe over using a screw? So yeah, there is. So if you would like to think of a ground electrode implanted in the brain, kind of the same way as like a microphone in audio recording, kind of getting the room noise, right? The room levels, if you will. So you get fluctuations maybe due to blood flow, as Sylvia mentioned, maybe some of these artifacts, but essentially it helps you parse apart how local your local field potential is, right? Because the local field potential is all of these inputs, everything that's not local coming to your recording site. So using different grounding approaches, basically comparing your recording to referencing it to either an implanted electrode or a ground screw, which is not making contact with the brain, can help you sort of parse these things apart. Okay. Let's see, we have maybe a question for Sylvia here. Is it feasible to do Neuropixels recordings alongside fiber photometry or miniscope recordings in another brain area? This is from Carolina.
I think yes, I don't see any problem with that. I mean, I guess the biggest problem is space. You need space for the Neuropixels plus for all your photometry. if you manage to do that, I don't think there should be a problem. Of course, I mean, if the light is really hitting the neuropixels, you might get the same problem as with optogenetics, but if that's okay, I don't see a problem. Yeah, I saw a question on switching recording sites. So you can do this with, depends on the software you're using, but with SpikeGLX, I think you can do this by really button presses. You just need to tell SpikeGLX which channels you want to record from, and you can just switch, and the switch takes less than a second.
Yeah, that's one of these benefits of having so many sites available is, yeah, just a click of a mouse and you're able to kind of bounce around to your different sites. Okay, Craig asks, can't you simply use shrink tubing to block the light out of the fiber optic cable? And I think this might be in reference to those
yeah, I'm not
artifacts.
sure I understand the question. So if I imagine you use the shrinkage around the neuropixels probe, then of course you also lose the contact to the neurons, right? I mean, you still need to see the neurons on your neuropixels, and the neurons need to see the light, right? Otherwise they wouldn't be inactivated or activated. So
I think, yeah, you might be able to get away with shrink wrap on the ferrule, the bit that attaches into the head, if you're worried about outside light.
That is true, but basically, so maybe this wasn't completely clear. I mean, the neuropixels probe is sitting in the brain, but the light is hitting the probe also inside the brain, right? So it's not outside the brain, and then inside the brain, you cannot shield the probe.
Right. Okay. Tal asks, do you see photoelectric artifacts during optostimulation?
So I guess I talked about this. I don't know when this, maybe the question came earlier, but
yeah, I think
so
you
yes,
did have.
if you do a step change in light, then clearly you see it. And yeah, also when you do these smooth changes in the light, like the raised cosine, you then see it in the low frequency band, so in the LFP band. So depending on what you're interested in, you need to think about what's best for your application.
Yeah, let me see. Craig got back to us here. Thanks, Craig. He meant to block the light coming out of the fiber to prevent artifacts, right? Yeah, so the light being inside the brain can't quite block it, but you can modify the optogenetic trigger itself, the shape of that. Okay, we have, can we record LFPs simultaneously with the multi-unit activity that Neuropixels allows you to record? So can you get LFPs with spikes with Neuropixels?
Absolutely. So actually the current version, Neuropixels 1.0, separates the signal into two bands. So one is 1 to 300 hertz. The other one is 300 to, I don't actually know what the cutoff frequency is, but several kilohertz. Just because the digitization rate is relatively low, so you have only 2 to the 10 bits. But if by separating this is then enough to get a reasonable digitization of the signal. But yeah, so you have all the frequencies. So you have LFP and MUA. In the new version, the Neuropixels 2.0, the digitization range is much higher. So 2 to the 16 or so. So you just get out one signal. And yeah, of all the frequencies you want. Of course, up to the Nyquist frequency. But the rate, at least the rate that we used was like 30 kilohertz. So yeah, that's plenty usually, even for the shortest spikes.
Absolutely. Okay, we have one more from Sinkyu. It says, is it feasible to get conduction velocity when the retinal cortex is recorded? So by stimulating the eye and recording the cortex, like a visual stimulus evoked potentials. So maybe I think Sinku is asking about with correct synchronization, right? You get the time delay between a flash and then the retinal cortex response, if I'm understanding that correctly. So conduction velocity.
About the cortex part, but I mean, yes, for example, you could just put a probe in the optic tract with all the axons and then record the LFP, and you should see the visual response, yes. And this has been done before, because I mean, it's still tricky to put your probe in, but to record an LFP in the optic tract has been done a long time ago in monkeys, so that's totally possible. The tricky bit really is to get to single unit activity here.
Mm-hmm.
And yes, you could also, of course, put the electrode in the cortex and see the visual response, like a visually evoked LFP.
Yeah, absolutely. And that's maybe one benefit to having multiple probe sites kind of feed into the same data acquisition, is that you know they're going to be synchronized in time. So you can do some of those. Okay. Oh, we have one more question rolling in. It can catch the small fiber neuropathy in NCS. Sorry, I'm not sure what NCS is off the top of my head here.
So is this maybe the person can come live? I'm not quite sure. So is this about damaging neurons?
Maybe, yeah. Knife, can you help us out here? What is NCS?
I guess central, or maybe CNS.
Maybe there's some
central nervous system. I guess. And,
maybe small fiber neuropathy.
Yeah, I mean, if it's about damage, I don't know any quantification. Yeah, I don't know of any study who has like measured cell death after neuropixels, a nerve conduction study.
Yeah, or perhaps might be able to parse some things out in the histology afterwards if you zoom in with high enough magnification. If your tissue is fixed, you may be able to do some antibody staining to mark maybe glia or some markers of damage of interest to you. It's another way to go about it.
Yeah.
Okay.
I think I can't add anything.
Great. Well, we wanted to thank everyone again so much for your time. And we really appreciate chatting with you. Yeah, please feel free to email us. And thank you so much for the really interesting questions. This has been a treat.
Yeah, great. Thanks a lot.